Hey everybody, thanks for being here today. I'm going to explain a topic to you today that I've had in my head for a long time, and that's action potentials. Action potentials is how an electric signal travels down a neuron. The other half of neuron communication or nervous system communication, synapses, we'll learn about next time, but this is how the signal travels within a single neuron before it passes the signal to another neuron using a synapse. Now, to cover this, we'll first talk about what resting membrane potential is and how ions are distributed across a membrane in a neuron. I hope it's all clear enough, and I hope you enjoy learning about what is going on inside your nerves. Now, before getting into the action portion of this, we're going to first look at how a cell membrane is for a neuron when it is at rest. That's called resting membrane potential. The membrane of a neuron, when at rest, actually is a little bit more negative inside and a little bit more positive on the outside. This is due to a few factors. One of those being that there are many proteins inside the cell, and proteins are generally negatively charged. There are large ions like sulfate, phosphate. Those cannot pass through the membrane, and those are typically higher concentration inside the cell. And there's a protein on the membrane, on the surface of the cell, called a sodium-potassium ATPase, or sometimes called a sodium-potassium pump. What that does is it uses energy from ATP to pump out sodium ions and pump in potassium ions. Those are both positively charged, but the idea here is that the sodium-potassium pump will pump out 3 positive sodium ions, but only pump in 2 positive potassium ions. And that causes there to be an imbalance of charge. You're pumping out more positives than you're bringing in. There's also, in the membrane, something called potassium leak channels, and that lets the potassium leak out of the cell. If you're pumping so much potassium into the cell, then there's a lot of it there, and it will diffuse out because it's higher concentration in and lower concentration out. And so if it has an opening, those leak channels are open all the time, and it can pass through because it's higher concentration in the cell and lower out. All of those things factor into the fact that the cell membrane is more negatively charged on the inside and more positively charged on the outside. Specifically, that is a negative 70 millivolt difference. The reason it's negative is because it's calling the inside more negative than outside. Now, here's a diagram that I've created to explain those concepts that I just went over verbally. This is the cell membrane. This is the inside of the cell, the cytosol. This is the extracellular fluid, the outside of the cell. I have a key here that I've got some sodium ions as green pluses, potassium ions as blue pluses, and in general, negative ions, whether they're phosphate or proteins or sulfate, will just be a red negative. I took care to put specific amounts of each of these on here. So notice the blue pluses, the potassium ions, are in high concentration inside the cell, and there are a lot of sodium ions on the outside. This half blue, half green protein is in the cell membrane, and that's the sodium-potassium ATPase. It is pumping out the sodium and bringing in the potassium. This is a potassium leak channel, so the potassium, because it's in such high concentration here, is going to leak out. You can also see that there are more negative charges here and fewer outside. And all that means that the cell has a potential to send an electric signal. So we'll look at how it actually sends an electric signal now that the potential is there. This is best explained with a set of diagrams, and I've created kind of a key here to explain what you're going to see in the diagrams. First, the action potential, it's the electric signal that goes down the membrane of a single neuron. It doesn't go between neurons, that's a synapse, and we'll talk about those next time. It's the signal that travels along the surface of the neuron. Now, there are a few key players in this. The other proteins that were mentioned about the resting membrane potential, those are still there, but will be largely omitted for clarity. There are sodium ions that take part in an action potential. there are potassium ions, and so I'm going to keep the same color coding scheme. What this is that you'll see is a voltage-gated sodium channel. Voltage-gated means that it opens and closes in response to different voltages. It opens if the voltage goes from -70 millivolts, which is resting, to around -50 or -45. And when it hits that voltage, it will open up, and because it's a sodium channel, it will let sodium ions pass through. There are also voltage-gated potassium channels, and those are open at positive 30 millivolts. If the electric potential at that part of the membrane is positive 30, then these will open. And they close when it gets back down to -70. An important difference between these, the way that they are diagrammed, is that a voltage-gated sodium channel has an extra little piece. So in addition to opening and closing the normal way that a ion channel does, it has this piece that can go in, and that's a closed and inactive state. And we'll see that in a moment. Now, I've chosen to break this down into 7 steps. Depending on the resource that you research, you'll find more or less. Sometimes steps will be lumped together. Sometimes one of these steps will be expanded into a couple of sub-steps. So I've started first at how it will be at rest. And again, this is the resting membrane potential with some things omitted for clarity. The key is that at rest, there are more sodium ions outside and there are more potassium ions inside. The resting membrane potential is -70 millivolts. And I've very carefully put numbers of positive potassium and positive sodium in the correct places. So I've got 8 of the sodiums on the outside, but only 6 of the potassiums on the inside, which means that it's more positive out here and less positive here. The negative charges are not shown, but still know that it's -70 millivolts inside the cell. I've also got 2 of each of these ion channels. They are both closed initially. The reason I've got 2 is we're going to talk about how an action potential happens here and how it can cause an action potential to then travel down. The overall message is being sent in this direction. I'll move on to the initial depolarization. Depolarization refers to the fact that it's getting closer to 0 millivolts. Polarized means that there is a voltage, and depolarized means that it would get closer to 0. So this initial or early depolarization is because somewhere up here, some sodium has gotten in and it kind of diffuses through. So now we've got a little bit more positive here and there's less difference, and it opens, or it triggers, the voltage-gated sodium channel to open. Next is full depolarization. The sodium ion channel that's open will let sodium ions in. The reason they would come in is for 2 reasons. First, they're positive and it's more negative inside the cell. Second, there are a lot of sodium ions outside and very few inside, and so they diffuse from high concentration to low concentration. That happens very quickly, and because there were more sodium ions outside, they all rush in and cause it to be more positive inside the cell right now, around positive 30 millivolts. So now we'll look at what happens. We've gone from -70 millivolts, we've gotten to the triggering point, and then we've let in a whole bunch of sodium, and now we'll see what happens at that top, the peak of the depolarization. Now, at peak depolarization, where it's positive 30 millivolts now inside the cell, so it's actually more positive inside, whereas it was the opposite at rest. At positive 30 millivolts, that triggers 2 important things to happen. The voltage-gated sodium channel closes, but not just closes, it closes and inactivates. This also will cause the potassium channel to open, and the potassium ions are going to flow. So you can see the difference here. This is closed and inactive. This is closed but could open. Next, because the sodium ions cannot come through anymore, but the potassium ions can leave, potassium is still very high concentration inside the cell and low concentration outside. So as soon as that potassium channel opens, the potassium ions are allowed to leave, and they will leave. Potassium ions are leaving, and that causes the inside of the cell, the potential to go back down and actually go even under -70 millivolts for a brief period. Propagation refers to the fact that some of the sodium ions that were let in this part of the membrane actually travel this way and cause this to open, just like we saw in the initial depolarization. And that means that this signal is going to go from this area to this area, and then we can assume that it would go here and here and here and travel down the axon, or travel down that part of the neuron. And last, I've discussed the steps that need to be taken for the neuron to reset, or for that part of the membrane to reset. What needs to happen is that the voltage-gated sodium channel needs to go from inactive back to active, and the sodium-potassium ATPase, or the sodium-potassium pump, needs to work very hard to restore the ion balances, that the sodium ions need to be outside the cell and the potassium ions need to be inside the cell. And that will make sure that it is reset and ready to send another signal, another action potential. Well, that's action potentials in a nutshell. I hope it was clear enough. Thanks for being here. I'm glad that you made it all the way through. And I hope that you've had as much fun learning about action potentials as I have had teaching you about them. I'll see you next time, where we will learn about saltatory conduction as well as synapses.